Welcome to another edition of CCSD Living Environment. The last time we talked about mitosis and how cells actually divide and the process that they undergo. So what we're going to look at today are some controls on the cell cycle, controls on that process, because obviously it needs to be controlled somehow. So how do they do it? Most cells divide until they come into contact with each other. Contact generally causes cells to stop dividing and growing. So what do we mean by that, and what would cause them to no longer be touching? Well, if somehow cells got damaged or some event took place that actually separated them, what will happen is there will be division that takes place to fill that space back in. And once they start touching again, then that division stops. So they get a signal from the other cells around them saying, hey, we're touching, we don't need to divide anymore. If the cells are no longer in contact, they start to divide again. So if they come back apart, they'll start the process over again. So what is that telling us? It tells us that there are controls on cell growth and division that can be turned on and off. So the same way with gene expression, we can turn on when a gene is going to be expressed, or we can turn off when a gene is going to be expressed. We can turn on when a cell is going to divide, and we can turn off when a cell is going to divide. And you can see where having the ability to turn those things on and off is a good thing. Regulation of the cell cycle in a eukaryotic cell. Remember, a eukaryotic cell is a cell with a nuclear membrane, like us. So regulation in eukaryotic cells is done by a group of proteins called cyclins. So cyclin is a protein that controls the cell cycle. Pretty easy to remember. And from there's many proteins that help actually regulate the cell cycle, both inside and outside of the cell. And we call those proteins regulatory proteins. Again, pretty easy to remember. Internal regulators are going to respond to events that occur inside the cell. The cell cycle can occur only if specific events have occurred inside the cell. For example, it will only go on if the DNA has replicated during the S phase of interphase. Anaphase can only continue if the chromosomes are attached to those spindle fibers where they can actually separate and move inside the cell. External regulators respond to events that are going to occur outside of the cell. The cell cycle is either told to speed up or slow down based on those external factors. And one of the things is something called growth factors, and I'm sure you can imagine that a growth factor is going to stimulate the growth and division of a cell. Remember, for our purposes, if our cells are going to undergo this process, then it's either because we're replacing something that's been damaged or we just want to grow and become larger. So this process for us, those growth factors, are actually telling the cells that they want them to divide. And that's because, obviously, growth is because we've added more cells. So what about these cells? Obviously, we know if a cell is damaged, we want to get it out of the way. But what if the cell isn't damaged? Do you ever think that there's a case where the cell might be destroyed? And there is a case, and that process is called apoptosis. Apoptosis is what we refer to as programmed cell death. So the cells have been given a signal to undergo this series of controlled steps that actually leads to the destruction of that cell. Now, obviously, it makes sense if a cell is tired or old or no longer doing its job that, okay, we want to get that cell out of there. But when else might we want apoptosis to take place? Well, in the development of our embryo, there was a bud of cells that basically was the forerunner to what we consider arms at this point. And the same thing was true with your hand. So what happened was, where all of those cells were filled in, we had to get rid of them. And apoptosis is what happened so that we could get those cells out of the way to give us the digits that we see now. And there's other structures in our body. Think about some other structures that you may have that could have been caused by apoptosis, because we wanted to get some of those cells out of the way that didn't need to be there. So it's a programmed cell death. The cells are being told, okay, you don't need to be there, we're going to get you out of the way. So that kind of takes us through the conversation about controls on cell division. And as I'm sure a lot of you have been thinking when we started talking about this, what if it's not controlled cell division? So uncontrolled cell division that we all know is cancer. In the case of cancer, we have body cells that lose the ability to control their cell growth. The consequence of uncontrolled cell growth in a multicellular organism like us is very severe. And unfortunately, many of us know people who have had cancer or who have had to deal with what goes on with the cells when they have cancer. So what is it? What is actually happening at the cellular level? Cancer cells don't respond to the signal that regulates cell growth. So as a result, the cells divide completely uncontrollably. When they're actually being told, stop dividing, they don't listen. They just keep dividing and dividing and dividing. So one cell becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, 16 becomes 32, 32 becomes 64, 64 becomes 128, 128 becomes 256. You can see how it just keeps going and going and going. And each time, it's just getting bigger and bigger and bigger and bigger. That's the problem with cancer, is that even though they're touching and that should be enough to tell them to stop, they don't listen to that message. Either because they don't want to or they just don't hear it. Whatever the reason is, they don't listen. And so that group of cells just keeps dividing. And that's how, even though it starts as one cancerous cell, it becomes many, many cells. So those cancer cells form a mass of cells that we call a tumor. Here's the thing about a tumor. We have two different options for a tumor. One case, it's not so bad, and in the other case, it is. And so what we need to look at is, what's the difference? In what we call a benign tumor, the tumors do not spread to surrounding healthy tissue or to other parts of the body. A benign tumor means that, yeah, those cells didn't respond to being told to stop dividing, but what they formed isn't necessarily a bad thing. Sometimes people go in and the doctor says, well, we have a mass of cells, we have a tumor, but it's not cancerous. So if you have to have a tumor, if you have to have a mass of cells, these are the ones that you want. Not to say that someday maybe they couldn't become a cancerous tumor, but for right now, what they've checked out, it's okay. It's not going to do anything. Those cells just didn't respond, and they're not doing anything differently. So that one's okay. The problem comes when we talk about malignant tumors. With a malignant tumor, the tumor invades and destroys the surrounding healthy tissue, and these are what we're going to consider a cancerous tumor. What starts to happen is those cancer cells start to take the nutrients and the oxygen and all the other good stuff away from the other cells and use it for themselves. And you can see where that starts to cause a problem, not to mention because they're getting all those nutrients and oxygen, they just keep growing and dividing and dividing and dividing and becoming bigger and bigger and bigger. So what do we hope for? We hope that they can catch those malignant tumors in time, and the doctor can go in and cut them out, or maybe they can give you chemotherapy, where they're putting chemicals in your system to kill those cancerous cells, or they can expose you to radiation, again, that's hopefully going to kill those cancer cells. And that's how we try to take care of those malignant tumors. And a lot of times, and as technology gets better, we do better and better at taking care of malignant tumors. So it's getting better. Still not a good thing to have, but it's getting better. There's a lot more hope. So malignant tumors, you say, well, okay, it's bad, but there's things we can do. Surgery, chemotherapy, radiation treatments, so we can try to take care of the problem. And that's true. So what's the issue then with a malignant tumor? The issue with a malignant tumor is when it undergoes a process called metastasis, or if they say the cancer has metastasized. What does that mean? Metastasis means the cancer cells spread to another part of the body and it forms a secondary tumor in that location. So for example, let's say that there was cancer in someone's large intestine and one of those cancer cells moved into the bloodstream. Where could that cancer cell go once it's in your bloodstream? Right, it can go anywhere in your entire body. So it comes off in the large intestine, travels through the bloodstream, and let's say it ends up in your lung or your heart or your brain, wherever it decides it's going to come back out of the bloodstream, and then what does it do? It starts dividing and dividing and dividing. So now that cancerous cell has moved from one location to a completely different non-related location, and that's what they mean when they tell you the cancer is spreading. doesn't necessarily mean that the tumor is getting larger. What it means is that it's traveled from location A to location B. Unfortunately, when tumors metastasize, it's very difficult for them to track where those cells are going or where they've actually gone. Cancer is caused by defects in the genes that regulate cell growth and division. What can cause those defects? Well, smoking or chewing tobacco we know can. Radiation exposure, whether it's ultraviolet rays from the sun or X-rays because something happened to you. Even viral infections. There are certain viral infections that can actually cause cancerous cells. In all cancers, the cell cycle control has been shut down. The idea that we can tell it to stop dividing, we can tell it, but it doesn't listen. And so those cells keep growing and dividing even though we don't want them to. And they've now found that many cancer cells actually show a defect in a gene called P53. What P53 normally does is it stops the cell cycle until the chromosomes have replicated like they're supposed to. Remember, during the S phase of interphase. But what they're finding is that for cancer cells that have a defect in P53, it doesn't do that. Whether they've actually replicated correctly or not, whether they've replicated at all, what it's allowing to happen is for the cells to divide, and they're not supposed to be. So that defect is one of the things that they're finding, okay, this is a problem in cancer cells versus other cells. So granted, it's not necessarily fun to talk about uncontrolled cell division, but it's something that we need to put out there and that you need to understand. We need to understand, how does uncontrolled cell division come about and what happens because of it. So next time we're going to move on and talk about other ways that cell division occurs, but for today, that's what we have. Thanks for watching the video. I'll see you next time.